Good afternoon, guys. So now it's afternoon. So now I'll show all the participants of the conference. So here, how we can see it. So here we can see all the list of students. So to make us sure that you're attended. So with the names and here nicknames. So don't forget to write your name in the chat. So in the final part, I'll explain. We will use it for assurance, so that you are in meeting. Okay, let's start the lecture itself. So the cell is a unit of health and disease, as we started before. So it's already fourth lecture. So let's continue our previous lecture. We stopped on that we have different types of receptors, and somehow these receptors are acting to our cells, and they change the cell consistency. We stopped on the growth factors, which control the growth of cells and growth of tissues, and facilitate the actual body size, body shape, and with help of them we do this. Okay, and the growth factor activity is mediated through the binding to specific receptors, ultimately influencing the expression of genes that can promote entry of cells into the cell cycle. So it's growth. So it's causing the hyperplasia. So relief blocks on cell cycle progression. So it also promotes the replication. Then prevent apoptosis. So when cells collect some injuries, in some day we can say so that this growth factor says us that it's okay to have damaged cells. So sometimes it's necessary to maintain the population of some cells, especially, for example, our RBCs. So still someone tries to add. Then and we can enhance biosynthesis of cellular components. So it's a process of hypertrophy. For example, some nucleic acids are produced, proteins, lipids, carbohydrates, which are required from mother cell to give rise to two daughter cells. That's that what is it. So this is functional growth factors and receptors to growth factors. Some growth factors are proteins that just stimulate cell proliferation and or survival. It is important to remember that they also can drive a host of other activities, including migration, differentiation, and synthetic capacity. And a little bit about this we discussed in previous lecture. Growth factors can be involved in the proliferation of cells at steady states, as well as after injury, after irreversibly damaged cells must be replaced. So also discussed. But sometimes, so as I noticed before, so some growth factors may activize depress the apoptosis process. So they depress the death of cells, which should be dead in other conditions. Sometimes it causes the tumorigenesis. Tumorigenesis is formation of benign or malignant tumors in our body. So because we suppress the apoptosis, we suppress the mechanism by which so usually our body handles with the tumors, and because of some other activity. But later it might cause causing the cancer. Okay. So that's this diagram I already shown you in previous lecture. So it's in the end of lecture I showed the growth factor activates the receptor. So here's a phosphorylized receptor causing the activation of RAC, and GTP. And works on deactivation of the active RAC. Then active RAC, so activize the cascade of reactions with causing the activation of transcription of some of MYC protein, and which causes the cell cycle progression. So we'll discuss about the cell cycle progression uh, furtherly. So guys, please in chat say to me, is this lecture visible? So do you see what I showed to you? This diagram. In chat, please type it in chat. Is it visible? Okay, let's continue. Okay, visible. I mentioned that it's visible, and then you see this cascade of reaction causing the activation of a MYC protein, which causes the cell cycle progression. And due to the cell cycle progression, so cells are produced in larger amounts. So then epidermal growth factor and transforming growth factor alpha, both of these factors belong to the EGF family, epidermal growth factor family, and bind to the same receptors, explaining their shared biologic activities. So both of them were working almost similar. They're produced by macrophages and variety of epithelial cells, and they mitogenic form hepatocytes, fibroblasts, and host of epithelial cells. So EGF receptor family includes four membrane receptors. Okay, that's not so important, but main idea is that these growth factors facilitate active growth of different cells. Growth of different cells. Once again, it's a hepatocytes, fibroblasts, epithelial cells. So that's it. Then we have a specific hepatocyte growth factor. Hepatocyte growth factor is a scatter factor. It has mitogenic effects on hepatocytes and most epithelial cells. So they are act as morphogen during embryonic development. Especially in embryology, they influence the pattern of tissue differentiation and promote the cell migration. So don't think that hepatocyte growth factor is responsible only for hepatocytes, because it was invented when it was found as a hepatocyte growth factor, but then afterward, after the investigation of it, we found other activities, other functions of these growth factors. Okay, platelet-derived growth factors. Platelet-derived growth factor is, according to name, we can see that it's produced. So these factors, they are proteins which activized by platelets. And when platelets aggregation happens, so one of the first activity of this platelet-derived growth factor is to activize that. So to activize the endothelium progression and to heal this part of blood vessel damaging. So having intrinsic tyrosine kinase activity induces fibroblasts, endothelial, and smooth muscle cell proliferation. So fibroblasts and endothelial cells and smooth muscle cells are components of blood vessel, and platelet-derived growth factor controls their growth and keeps the so it's necessary to heal the damage of blood vessels. So, and thus promoting recruitment of the cells into areas of inflammation and tissue injury, as I said here right now. Vascular endothelial growth factor, also very interesting factor. So one of the main factor you should remember, it's VEGF-A and VEGF-B and C and D and placental growth factor is a family of homodimeric proteins, so generally referred to simply as VEGF. VEGF-A is usually named as VEGF. VEGF-A is its most common type. It's this factor responsible for angiogenesis, including blood vessel development after injury and in tumors. So you see, functionally, it seems like very close to platelet-derived growth factor, but actually they work alone. The one works with another, and also, but this growth factor is activized not just to heal, but just to increase the amount of blood vessels in this area. In case of, so you remember that. So might be you heard about the inflammation. You know that in inflammation we have redness. It's it's one of inevitable features of this VEGF, so of inflammation to possess more blood vessels in this area, which causes the redness of this. And one of the causes of it, it's involvement of VEGF. So VEGF-A, especially too, causing the capillary sprouting, which causes the appearance like inflammation. Okay, vascular endothelial growth factor bind to a family of receptor tyrosine kinases, so it's highly expressed in endothelium and in the most important for angiogenesis. Antibodies against VEGF are approved for the treatment of several tumors such as renal and colon cancers because cancers require angiogenesis for their spread and growth. So one of tricks which our tumor cells do, so they produce this VEGF. So you know that when cells are growing very fast, so they require a lot of energy and oxygen for their activity, and so not too much oxygen, but more nourishment. They need building blocks. They need amino acids, carbohydrates, and even fat. They need so different lipids. So and that's why VEGF is produced by most of malignant cells. And due to this, a lot of blood vessels entered inside this and can nourish all the cells. So, and it's a bad effect of this, and that's why we use some drugs anti-VEGF antibodies, so which suppress this protein activation of receptors, and that's stopping the growth of blood vessels. And that's why most of so some tumors can die just because of suppression of VEGF. So also keep this option in treatment of cancer patients. Okay, extracellular matrix. So we didn't discuss the extracellular matrix in previous lecture. So let's discuss the function and then activity of this extracellular matrix. Function is mechanical support, so it's necessary for cell anchorage and cell migration and maintenance of cell polarity. So especially in epithelial cells, you remember that we mechanically support the tissues on the proper side. Control of cell proliferation. So extracellular matrix gives so it's not just media for cell living, yeah, but actually they bind by binding and displaying growth factor and by signaling through cellular receptors of the integrin family. So you remember integrins are the molecules which bind the extracellular matrix with intracellular matrix, and rupturing of them causing the activation of growth factors, and some provides a depot for a variety of latent growth factors. So these latent growth factors which happen when tissue injured, so it activates the proliferation potential of the cells. Then another function of connective tissue, extracellular matrix, is scaffolding for tissue renewal. So its maintenance of normal tissue structure requires a basement membrane or stromal scaffold. The integrity of the basement membrane of the stroma of parenchymal cells is critical for organized regeneration of tissues. So scaffolding, so just one word explains this well. Those extracellular matrix disruptions result in defective tissue regeneration and repair, for example, cirrhosis of the liver resulting from the collapse of the hepatic stroma in various forms of hepatitis. This we're going to discuss in, I guess, in next semester in digestive tract pathology in this part. So, but still keep in mind that connective tissue and too much extracellular fibers is sometimes is very bad. Establishment of tissue microenvironments. The basal membrane acts as a boundary between the epithelium and underlying connective tissue. It does not just provide support to the epithelium, but is also functional, for example, in the kidney, forming part of the filtration apparatus. In histology or usually in histology or in physiology, you discuss these features, functions of this connective tissue. So I don't need to stop here for a long time. So here, what I talked about. So you remember focal adhesion complexes, there are hemidesmosomes, which connect the intracellular matrix, which is composed of actin cytoskeleton, and extracellular matrix. With help of intergenes, we connect the cell with fibronectin and collagen. Okay, and disruption here may activate the growth factor receptors, which gives information to the cells to produce something new or to split the cell. So it doesn't matter. So but anyway, so it's not just silent connection. If this connection is active, and when this connective is damaged, it can cause a proliferation or differentiation, protein synthesis, attachment to something, migration, or even shape changes. So that's activity of extracellular matrix out of you used to think about it. Extracellular matrix is constantly being remodeled. Its synthesis and degradation accompany morphogenesis, tissue regeneration and repair, chronic fibrosis, and tumor invasion and metastasis. Extracellular matrix occurs in two basic forms: interstitial matrix and basement membrane. Interstitial matrix is present in the spaces between cells and connective tissue and between the parenchymal epithelium and the underlying supportive vascular and smooth muscle structures. So you know it, so no need to discuss. And basement membrane, so it seems like a random array of interstitial, random array of interstitial interstitial matrix in connective tissues become highly organized around epithelial cells. So endothelial cells and smooth muscle cells forming the specialized basement membrane. So it's made by in easy words, we can say that it possesses non-fibrillar type 4 collagen and laminin. So type 4 collagen forming the basal lamina, and laminin forms the reticular lamina. And fusion of them together, so as part of epithelial cells and part of connective tissue, forming the basement membrane. So, really, that's it? No, it's not it. So it's damaged type. So now I'll continue this lecture. So must be some mistake happened. Okay, let's continue. So here we discuss the interstitial matrix and basement membrane. So which components of extracellular matrix we should mention? So I continue with the book. So it's you have this book. My task is to explain you this book. So sorry, I'll show you this book. So yeah, components of extracellular matrix. Components of extracellular matrix fall into three groups of proteins. It's fibrostructural proteins such as collagens and elastins that confer tensile strength and recoil. So I hope you remember. Then water-hydrated gels such as proteoglycans and hyaluronan that permit compressive resistance and lubrication, and adhesive glycoproteins that connect extracellular matrix elements to one another and to cells. So let's talk about collagens. So here it's a basement membrane. I said yes, we have type 4 collagen, laminin, and between them we have proteoglycans which bind them together. Collagens. Collagens are composed of three separate polypeptide chains braided into a rope-like triple helix. So we have about 30 collagen types. we have most common types of collagen like types 1, 2, 3, uh, until 5. So we already discussed what is 4. Yeah, it's present in cell membrane. Um, sorry, in a basement membrane. So let's continue. They are cross-linking. They cross-linking triple helices by covalent bonds and unusual post-translational modification. Modification that requires hydroxylation of lysine residues in collagen by enzyme lysyl oxidase. So this lysyl oxidase is necessary. I will show you here. This lysyl oxidase is necessary to change the shape of this protein. So change the shape of this protein to interconnected parts. So for example, in elastin, especially in elastin, um, and also there. This is necessary to form these binds. So I'll show it closer. So here in collagen and elastin, we have bridges from one fiber to another fiber, and these fibers are possible only with this elastase, only by the lysyl oxidase. This lysyl oxidase is controlled with vitamin C, and that's why we have this disorder which is named as scurvy. Yeah, when first of all connective tissue suffers, and no proper connection between one fiber with another fiber exists. And that's why we feel weakness in teeth, periodontal spaces, yeah, and blood vessels get very easy to rupture. So due to in vitamin C deficiency. Okay, and we have non-fibrillar collagens which don't form fibrils, so contribute to the structural or planar basement membranes. It's in type 4 collagen, as we discussed right now. Help regulate collagen fibril diameters of collagen interactions via so-called fibril associated collagen with interrupted triple helices. So anyway, so let's move forward to elastin. Elastin is also it's collagen like protein, so it's possible to get these changes. So when it's relaxed, so it forms something like coils which are connected to each other. So that's main opportunity of elastic fibers. But when we stretch it, they became straight but still connected. This feature is used in our elastic arteries like aorta and large arteries, and also in muscular arteries. So they have a lot of elastic fibers, which allows them to stretch and relax along with the pumping of heart. Also, this elastin is present in our elastic cartilages. It's present in skin, in dermis of skin. So in which keeps us in stretch position. So let's talk about proteoglycans and hyaluronan. Yeah, sorry. So in individuals with Marfan syndrome, so with this fibrillin defects are present. We will discuss in genetic disorders and pathophysiology of these effects of Marfan syndrome and pathological anatomy of this Marfan syndrome. We will discuss a little furtherly in topic of genetic disorders. Fibrillin controls the availability of TGF beta, transforming growth factor. So let's discuss later. Then proteoglycans and hyaluronan. Proteoglycans form highly hydrated gels that confer resistance to compressive forces in joint cartilages. Proteoglycans also provide a layer of lubrication between adjacent bony surfaces. So it seems like it's some jelly component in our body. So and it makes the gel-like components in our body. So in all connective tissue, uh, but more evident for it is in cartilages. Okay. but they also produce adhesive glycoproteins and adhesion receptors. Let's talk about them. These are structural diverse molecules variously involved in cell-to-cell, cell-to extracellular matrix, and extracellular matrix to extracellular matrix interactions. Prototypical adhesive glycoproteins include fibronectin, a major component of the interstitial extracellular matrix. So, okay, let's not stopping on this too much. So we have composition of fibronectin is here. So you see disulfide bonds here. So we have fibrin binding domains, which are and they in okay. It's complicated biochemical molecular biology stuff, but main idea is that it binds the extracellular matrix to intracellular matrix, and proper connection of this is affected to changes inside the cell. So actually, I already said about this. Okay, it's not the point of today's discussion. Later we'll discuss it in a better way. Next chapter is maintenance of cell populations, proliferation in the cell cycle. I hope I don't need to remind you the cell cycle, but still we must to know about it. So mitosis, you know the mitosis, that in the end of mitosis after the telophase, there are formed two daughter cells, and they start to come into G1 phase. G1 phase is a growth phase in which cell gains more proteins, more ribosomes, more organelles, and so everything is normal activity of cell. So what we used to think when we talk about cell. So it grows and it collects something, it gets some activity, and some of cells. So actually most of cells, after the cell division, they don't undergo another division. It undergoes into G0 phase, when this phase in which the cell is resting for a stimulation. So for stimulation, for example, by growth factors. And when growth factors activity actually acts on G0 phase to continue the migration of cell along the cell cycle. Then here we have the restriction points. In these restriction points, okay, we'll discuss these restriction points. But main idea of checkpoints along the cell cycle to prevent damaging cells to undergo cell division. We have a restriction point check for DNA damage. It's G1S checkpoint, one of the main checkpoints, because during the mitosis might appear some mistakes, and we need to check them out. And if they are damaged, so it might cause the cancer after all. So we need to suppress the cells. Okay, later we have check for damaged or unduplicated DNA. So after the S phase, when some DNA is not properly duplicated or might be erupted due to some chemicals or radiation or any other injury type, so we have to check it after this. So and then finally we have mitosis. So let's discuss this. The cell cycle is regulated by numerous activators and inhibitors. So I'm not going to explain every type of this, every part of this. So we'll better discuss it in tomorrow establishment. So, but what you need to remember is that we have cyclins, special proteins which control the cell cycle, and but later will be explanation here. So you see, we have cyclins which activate the CDK, so like a receptor, like a protein activates the receptor, and we have a special inhibitors of this. And okay, let's start with G phase. Yeah, cyclin D activation activates the CDK4, and it leads to transport the cell into the S phase. And in S phase, activation of different cyclins, cyclin A here also, they can pass it out. But if these cyclins are not produced, if the cyclins are not attached to CDK, so which it is happened due to CDK inhibitors, promotion of the cell cycle is erupted. And furtherly, in apoptosis, I'll explain one of mechanism how to disrupt the cell cycle by cell death. So when some cell which with potentially damaged DNA keep the cell cycle continuation leads to the death of the cell. That's activity. 30 minutes of lecture passed, and someone wants to join. Sakib Hanif Maitla. Not bad. Okay. Under conditions of homeostasis, stem cells are characterized by two important properties. So, what are stem cells? It's undifferentiated cells which are able to renew the cell population in this area. So, and function is self-renewal, which permits stem cells to maintain their numbers. Self-renewal may follow asymmetric or symmetric division. So, cells might be divided symmetrically or asymmetrically. You have a lot of examples from histology about it. So, for example, asymmetric divisions refers to cell replication in which one daughter cell enters a differentiation pathway and gives rise to mature cell, whereas the other remains undifferentiated. So, best example of this is so it's present in testes of males. So when spermatogonia A spermatogonia A is not transported into to spermatogonia B. Spermatogonia A produces two cells, one of which is spermatogonia A, another one is a future spermatogonia B. So that's an example of this. Typical stem cells. And we have different stem cells like embryonic stem cells. So they are most undifferentiated. They are totipotential stem cells, so which are able to produce all the cells, every cell in human body, if we make them, if we add their proper growth factors and some activation. So that's it. We have baseline cell population. And baseline cell population is renewed by their own activation, but they can proliferate and increase their mass. It's a typical stem cell population. They can differentiate themselves into other type of cells. So we'll discuss it in some topic. This baseline population is renewed with help of stem cells. So that's always we have some a pool of cells which are producing these cells. For example, in the mucus of respiratory tract, we have a lot of stem cells which are resting near the basement membrane, while other cells are located in upper side. But also some baseline cell population may undergo the apoptosis. So we can that's a mechanism of regulation of cell populations. So we can renew them to produce more cells. We can kill them, and we can even if the entire cell population is died, we can renew them with help of stem cells. And this cell population might be differentiated in some other tissue type. So, and this is how stem cells are produced. So zygote is a typical totipotential stem cell, so which is able to produce all parts of embryo, so including the inner cell mass and outer cell mass, so which is also known as trophoblast and embryoblast. Then these cells, these four cells which are shown here, they are known as pluripotent stem cells. They are responsible for formation of our human cells, like endoderm-derived cells, mesoderm-derived cells, and ectoderm-derived cells. So, and they can renew themselves or producing the next level of differentiation, like multipotent stem cells. You know these multipotent stem cells, they are present in our in blood formation period. So they produce different lineages of cells, for example, myeloblasts, yeah, for example, also lymphoblasts, which later will be differentiated into mature cells. Actually, we are going to use these pluripotent stem cells in medicine. So in previous year we have a conference. So from your batch no one acted. So that's why I'll repeat this thing. So some students prepared 2 or 3 reports about the stem cells and how we are going to use them. So potentially we can culturize the stem cells. So directly from you, yeah. So when you born out, we can save some of these stem cells. They are located in our umbilical cord, inside the umbilical cord. We can save them, freeze them, and in time when you need to replace some organ or replace some tissue, or need to heal something, we can use your stem cells to produce your own organs, like pancreatic islets. Might be so in cases of some blood disruptions, so we can produce hematopoietic cells. In cases of cardiac infarction, we can produce cardiomyocytes. In case of brain infarction, we can produce neurons, or nerve injury. Yeah, we can produce neurons from this culture of pluripotent stem cells. Or in cases of some organ failure, for example, liver failure or kidney failure, we can grow a new organ, entire organ for you, just to replace it. So during the and without risk of rejection, yeah, because it's your own cells. And so that's a very cool option, but now it's unavailable yet, but very soon I guess in this century it will be available. Okay. So what is so yeah, stem cells is still explanation of what is stem cell. And this is what's regenerative medicine. So, concluding remark. So let's talk about concluding remarks. This survey of selected topics in cell biology serves as a basis for our later discussion of pathology, and we will refer back to it throughout the book. So that will be in book and in lectures too. Sakib Khan so came in the last moment. Let's go. Okay, today I'm not going to explain more, so it's not real lectures. So I hope everyone who wanted to hear me heard me properly. So I'll use the same explanation, same video. We made it right now and load it in YouTube, and I share to your leaders. So ask them to give it to you if you need to confirm something. But I my first task to you, so please open books and read it by yourself too, because I skipped a lot of important points just because of less time we have. Okay, thank you for attention. So let's make a final checking of attendance. 60 participants. So Amir Fahad here, Abdul Jalal, Afak Ahmed Vaga, Sarukama. Okay, 63. Thank you. Goodbye. See you next time.